Thanks for joining me again. This is the starting with the Adelase gene expression, Xenopus laevis, which is that African clawed frog. This is started. It's part two of the presentation that was on the different isoforms of Adelases and their different expression patterns. And we've covered 1 through 10 slides. We covered Adelase and its a history, the different 3 types of Adelases, and now we're going to actually use an example of the African clawed frog. Like I said, we're going to go through the expression patterns of these different 3 types of Adelases on one species. So Adelases as materials for studies on differential gene expression. Adelasa fructose 6 bisphosphate adelase provides a typical example of developmentally controlled multilocus type isozyme. Its functional form is either homo or hetero tetrameric, composed of 2 or 3 different 40,000 Dalton proteins, A, B, and C, each coded by a different group of genes. Holoenzymes catalyze different reactions depending on the nature of the constituent subunits. Basically, Adelases A and C catalyze the cleavage of fructose 1,6-bisphosphate in the glycolytic pathways, giving rise to the glyceraldehyde 3-phosphate and dihydroxytane phosphates, whereas Adelase B catalyzes cleavage of fructose 1-phosphate derived from dietary carbohydrates to give rise to the glyceride and dihydroacetate phosphates. In addition, Adelase B catalyzes gluconeogenesis or the hexose formation. Interesting, the expression properties of Adelase subunit genes A, B, and C differ according to the specialized roles which they perform. Thus, Adelase A is expressed mainly in the muscles and the tissues with a high rate of glycolysis, and Adelase C is expressed specifically in the brain and nervous tissues. By contrast, though, Adelase B is adapted for dietary fructose metabolism and glyconeogenesis, is expressed in the liver, kidneys, and intestines, which are active in these pathways. As a result, mutation of this gene in humans causes severe hereditary congenital disease, fructose intolerance. There is interest in control mechanisms of expression of the 3 Adelase genes in Xenopus embryogenesis. It was asked which of the 3 Adelase genes are utilized in early embryonic development, during which an unicellular and totipotent egg cell cleavage rapidly and becomes a multicellular embryo composed of many differentiated cells. The cloning of the Xenopus Adelases A, B, and C clonal DNAs, the clonal and expression studies of these 3 Adelases were done, and it was obtained that the X-aldo-A, X-aldo-B, and X-aldo-C, it showed that amino acid sequences for these as a number of the amino acids reduced from clonal DNAs, there was no difference in all the 3 Xenopus Adelases. The overall identities at the amino acid sequence level between the Xenopus Adelases A, B, and C, and between B and C were lower, 67 and 68% respectively, than that between Adelases A and C, as in rats and in humans. Also, it was marked at the 4 isosome group specific regions, which are conserved in each group of Adelases A, B, and C, differently marked 2 of these, since these are particularly important for Adelase B specific function utilization of fructose 1-phosphate. Furthermore, it marks 7 regions which are highly or relatively highly conserved in all 3 types of Adelase sequences. In these 4 were considered different among the 3 Xenopus Adelases. With less than 50% identity, yet those in 7 conserved regions were similar between A and B, 85%, and B and C, 85%, and A and C, 93.8% in amino acid identity. Xenopus Adelases A, B, and C contains lysine at amino acid number 230 as an active center. The 3 Adelases have a similar 3-dimensional structure. The phylogenetic tree constructed by the neighbor joining method showed that the ancestral gene of Adelase B genes segregated from the ancestral gene of Adelase A and C genes before segregation of the A and C genes in Xenopus, as in any other animal species. It was obtained a cell line that the Xenopus adult kidney and fluorescence in situ hybridization studies showed that the 3 Adelase genes are located in different chromosomes. Now we'll continue on with talking about the African clawed frog. The expression of the three allelase messenger RNA in adult frogs. Northern blot analysis was done using RNA extracted from various Xenopus adult tissues and obtained a very strong allelase A messenger RNA signal in muscle, a moderately strong signal in heart, brain, and intestine, and a low level of expression in all the other tissues, probably due to the transcription from the minor pan-expression promoter. Allelase B messenger RNA appeared as a signal in kidney, liver, intestine, stomach, and interesting in skin, as noted for humans, rats, sheep, sea bream, and salmon. The expression in skin is as in salmon. We obtained a strong signal of allelase C, messenger RNA in the heart, brain, and ovary, and a weak signal in intestine, testes, lung, and other tissues. Using in vitro transcribed allelase A, B, and C messenger RNA as references, blot testing were done in calibration curves for determinations of absolute amounts of A, B, and C messenger RNA in adult tissue, then calculated the percent composition of these three, the total amount of three allelases messenger RNAs in A, B, and C plus C per unit amount is the largest in muscle and next largest in heart due to the occurrence of the extremely large amount of allelase A messenger RNA in muscles and allelase C messenger RNA in heart. This results in a reasonable in view of the fact that these two tissues are most active in the energy consumption. It is interesting here that these adult skeletal muscles express almost exclusively allelase A, but the heart cardiac muscle expresses much more allelase C messenger RNA than allelase A messenger RNA. Allelase A messenger RNA constituted as much as 99% of the total allelase messenger RNA in muscle, whereas allelase C messenger RNA constituted about 70% and 60% respectively of the total allelase messenger RNA in the heart and the brain. Thus, the percent composition of the three allelase messenger RNAs in the brain, a typical ectodermal tissue, is similar to that in the heart. A typical mesodermal tissue, allelase B messenger RNA, on the other hand, constitutes the major allelase messenger RNA in liver and kidney and also in stomach and intestine, again reflecting the fact that allelase B is actively expressed in tissues that are necessary for metabolism and dietary fructose. It is noteworthy that kidney, which is mesodermal in origin, has an allelase messenger RNA composition almost identical to that of the liver, which is endodermal in origin. In both of these tissues, allelase B messenger RNA constitutes about 85% of the total of messenger RNA, and the rest being allelase A, with only a trace of allelase C. In intestine and stomach, there's both endodermal, the percent composition of allelase B messenger RNA exceeded 50%, in skin, which is ectodermal, also strongly expressed allelase B messenger RNA, about the amount being slightly lower than 50% of the total allelase messenger RNA. Thus, each tissue has its characteristic allelase messenger RNA composition, thereby reflecting functional differences between the three allelases rather than differences in the developmental region of each tissue. Also, the northern blot analysis was made of the messenger RNA for the oocytes, the developing embryo. It was made using these RNAs from embryos at different stages and in vitro transcribed three allelase messenger RNA using the calibration curves to correlate strengths of these signals, amount of allelase RNAs. Here again, estimated the amount total of each of the three allelase messenger RNAs in embryos. The Xenopus fertilized egg contained approximately 60 parts of this, whereas of messenger RNA of allelase A, 45 parts was allelase C, and only 1.5 parts was allelase B. Thus, allelase A and C messenger RNA together constitute 99% of the total egg allelase messenger RNAs in the egg, and allelase messenger RNA constitutes approximately this amount of these as well. It also says from the early Neurula stage, however, the level increased sharply at the tadpole stage, stage 35. The period of this great increase in the amount of allelase A messenger RNA corresponds to the period of this great amount of increase in that number as well. The amount of allelase C messenger RNA per embryo also decreased sharply until the gastrula stage. Unlike that of allelase A messenger RNA, however, the post-gastrula increase in the amount of allelase C was not large, and the level remained at about the same throughout. The very low level of allelase B was maintained until the mid-gastrula stage. At the late gastrula stage, however, a slight increase was detected, and from the late non-rula stage on, the amount continuously increased and eventually reached the level that stayed as an adult. Based on the quantitative data, it was calculated that the percent of composition of these three atolases are messenger RNAs from embryos at different stages. It appeared that from A plus the C type atolase composition is egg-specific or nearly embryo-specific. Probably this atolase composition is suitable for fructose metabolism during the yolk-dependent developmental stage. And for 13, we're talking about the tissue-specific expression of atolase. It talks about in this, according to the whole amount in the pseudohybridization, the earliest stage we detected these local A, B, and C. In the neurula stage, 25 and 21 neurula stage, we're talking about the different stages of these developments. Throughout the latter stages, though, the atolase A messenger RNA signal was noted in somites. At the tail bud stage, the atolase B messenger RNA signal appeared in regions where liver rudiments and proctoderms were formed. After hatching, atolase B messenger RNA was strongly detected in the protophorous liver rudiments and hindgut, and at this stage, the tail fin epidermis was also stained. At stage 23, the atolase C messenger RNA signal was in a neural tube, and at stage 33, it was in the brain, spinal cord, so it continued to develop. In the dorsal view of the hatched embryos, the regionally different expression of the three atolases was clearly seen. Therefore, we started getting more of the atolase B as the organism started using more to bring in the food sources. And the genomic DNA structure for this, it talks about the genomic DNA cloning of atolase B was also obtained. A clone for this contains the entire sequence of the non-coding and the coding for this Xenopus gene. For comparison of the genomic gene for Xenopus atolase C, clonal DNA was found to consist of 9 exons. Xenopus atolase C gene closely resembles human and rat atolase genes at this point as well. However, the size of the gene as a whole was quite different, even though that they were similar between these. However, the size of the gene as a whole was quite different between the Xenopus and mammals due to the fact that all of the introns in a Xenopus gene are several fold larger than the human and rat counterparts. Also, the primary extension analysis was done using the oocytes, and it was also said the proximal part of this region was also, just like rats and humans, the Xenopus atolase C gene contained specific elements. Furthermore, the Xenopus gene at the CCAAT box and a TATA-like element were found, and these were not found in humans and rats. Let's go back to what we've talked about as far as this goes. The Xenopus laphus atolases expression in summary. In amphibian embryogenesis, it is a general phenomenon that a large amount of material stockpiles support early development, and these are replaced by materials which are newly formed. Based on the zygotic gene expression during development, such as a switch from the maternal to the zygotic messenger RNA, was also observed for the atolases A and C. The maternal atolase B messenger RNA occurs only at a residual level, and extensive transcription takes place only after the late neurula stage. This agrees with the zymogram analysis. Predominant enzymes present in the early embryos were atolase A and C. We confirmed that the tissue-specific expression patterns in embryos was mostly as in adults. Finally, one may ask why the atolase B gene, which is needed for metabolism of dietary fructose, is expressed so early at the gastrula stage during embryogenesis. In embryos, however, such a precocious activation also occurs in other genes. For instance, the transcription of the ribosomal RNA genes begins as early as the late blastula stage, despite the fact that the embryos contain ribosomes of an amount sufficient to support development up to the stage 41, which is feeding tadpole stage. Well, thank you for joining me. I'm going to go back through my references. There's a conclusion. We talked about the different topics of that, of what atolases are and the 3 different types. And we also talked about the African clawed frog, the actual example of that. And here's my references, the first 4 here. We have 4 more references here. Thanks for joining me. I hope you enjoyed the video. Hope you know a little bit more about the subject.